We're really excited to be able to have this session. I think it's been a long time coming to have multiple therapies in the space being developed for those with Becker muscular dystrophy. It's an underserved population, and so we're really excited to see that, as well as some of the other studies that are going on to help further our understanding of natural history of the disease. So excited to be able to hear some talks today. So a few great talks. We're going to go through the speakers, have their presentations, and then at the end we will have a Q&A with everyone. So once we do that, we have 2 mic runners here in the room, and we'll be happy to take questions from everyone. So our first speaker today is Joanne Donovan from Edgewise Therapeutics.
Well, good afternoon. It's a real pleasure to be here to speak to you in the room, and also aware that there's a number of folks online listening in as well in the Becker community. So today I'm going to speak about EDG 5506, which we at Edgewise have been developing as a novel approach for Becker muscular dystrophy. We have started in Becker. We are also developing it in Duchenne as well. So we are a public company. So, isn't this supposed to move? Okay, we are a public company, so I do need to show you this forward-looking statement, which says that, among other things, this is an investigational agent that is not yet approved in any country or territory, and I am an employee of Edgewise. So I'm going to start a little bit. This apparently needs 2 clicks. There we go. To tell you about an approach that is a little bit different than what you have been hearing about in many of the sessions to addressing the clear unmet medical need in Becker muscular dystrophy. And what the goal here is to protect the susceptible muscle fibers. And we know that there are one type of muscle fibers. Muscle is made up of 2 types of fibers. And we know that there is a particular type called fast muscle fibers that are most susceptible to damage, particularly the type of damage that happens with mechanical stress on muscles. And that's what we are aiming to protect. We have developed an investigational therapy, EDG 5506, that is designed specifically to protect these susceptible muscle fibers from damage and consequently protect the whole muscle. What we've seen so far is that in diseased animal models, EDG 5506 does protect these muscle fibers and prevents long-term development of damage in the muscles in MDX, as well as in the golden retriever model. So, and lastly, I'm going to show you some clinical data from the studies that are completed and ongoing in the clinic. Good, it only clicked. So I'm going to start back at a level and talk about what dystrophin does. So we essentially every session here, we hear about the role of dystrophin and that replacing dystrophin has the potential to address and improve the disease prognosis. So just to step back, well, what does it do? And dystrophin is not a protein that contracts muscle. It doesn't provide power to the muscle. What it does is it protects the muscle from stress during contraction. And the way it does this is by linking the muscle fibers so that they can all move in together to reduce the stress on any single muscle fibers. So this, that wasn't what I wanted to, I didn't even press that. Back, okay. So what this shows is the contractile mechanism of the muscle fiber. There's a protein called myosin that interacts with another protein called actin. And together, they create the power for the muscle. That is what directly contracts the muscle and provides the function, basically, to do everyday tasks. What dystrophin does is it links across muscle. It links that contractile apparatus to the next cell and through the interstitial fibers that are between muscle cells. So its role is basically to connect cell to cell so that when one fiber contracts, it distributes the force across the muscle. And when that is missing, then the mechanical stress damages the muscle fiber. It literally disrupts, when we look on electron microscopy, it disrupts the sarcomere, the basic unit of the muscle. And when dystrophin is lacking, of course, the mechanical stress that occurs damages the muscle. And what that consequence of that is it leaves it causes the muscle to be replaced with fat and fibrosis. So it's not the lack of dystrophin that causes the decreased strength. It's the loss of the muscle, ultimately. We know that boys in their first couple years, they can function normally with muscles that are completely lacking dystrophin. It's the effect of that dystrophin on preventing damage that ultimately leads to the loss of function. So what our approach is to modulate that mechanical stress. And this doesn't work the first 3 times. There we go. So what this does is to prevent the mechanical stress causing damage. And with the compound EDG-5506 is distributed to the muscle, then that can protect the muscle. It can protect the muscle when it's contracting. And I'm going to show you an example of this on the next slide here. Possibly. There we go. So what this shows is actually a finger muscle of an MDX mouse, the classic model for Duchenne. And what you see here on the left is the muscle contracting. And what you see as it continues to contract is that there appears to be little blebs that appear in the individual fibers. And then you see that they start to glom up, basically, and you see this very visible damage to the muscle. And those are presumably the fast muscle fibers that are getting damaged. And what happens over time is this creates irreversible damage to the muscle. On the right is what happens with EDG-5506. This is applied to the muscle before contraction. And there, the muscle from an MDX mouse is contracting, and you don't see the development of that kind of damage. So our hope is that by preventing that kind of damage, we can preserve function and potentially improve function, as we've seen in the MDX. Okay. Now, what happens when the muscle fibers are damaged is that they release muscle protein biomarkers. You've all heard of CK. You know that it's elevated both in Duchenne and in Becker. And what happens is when the muscle membrane is damaged, it allows the release of proteins that are inside the cell into the surrounding area that can be picked up by the bloodstream. And that allows us to measure in the bloodstream things like creatine kinase, CK, myoglobin, fast troponin, which reflects these fast muscle fibers, as well as a number of other proteins. And that allows us then to see what kind of damage is occurring to the muscle fibers. Now, after looking at this compound in MDX mice, in golden retriever models, we took it into phase 1 studies. And we asked, this is the first studies in man, we looked at both unaffected adults as well as those with Becker. There. And we asked some very basic questions in this study. We dosed for no more than 2 weeks. And what we're looking for in this kind of a phase 1 study is, well, what do we know about safety? What do we know about side effects? And with increasing doses of the drug, when dosed for 2 weeks, we saw that it was well tolerated. The most common side effects were transient drowsiness and dizziness, and both of these seemed to resolve after the patients were taking, or the healthy volunteers were taking, for a few days. So it also answers questions about how are we going to give this in future clinical trials. This is the first time we're in people. And so what we found was that as a tablet, that EDG 5506 is well absorbed. And the time course of how it's absorbed allows us to give it once a day, and give it once a day either with food or without food. And we asked a very important question about whether or not it's getting to the target, whether or not it's getting to the muscle. Because if a drug is going to work, it needs to work where it needs to be able to get to the protein, to get to the target. And what we saw was that EDG 5506 was concentrated 100-fold in the muscle, in a muscle biopsy, versus in the bloodstream. So that tells us that the drug is getting there, it's binding very tightly to the fast myosin, which is the target, and that's what we need to be able to have the drug work. So those are very important take-home lessons from the study. We also looked at biomarkers. Okay. And we looked at creatine kinase, and we looked at fast skeletal muscle troponin, which is very specific for the fast muscle fibers. The first bar on the left of each shows the levels in unaffected. CK levels are low. In fact, fast skeletal muscle troponin I is usually not even detected in unaffected adults. At baseline, before they started EDG 5506, the levels were high. We had these patients, these Becker patients, were very gracious to come into a phase 1 unit for almost 3 weeks, which was really asking an enormous amount of them. And we are very grateful to them for doing this. And in a phase 1 unit like this, they're less active. We actually measured this with a pedometer. They're not walking around as much. And that actually decreases your CK and your fast troponin. It reflects that you're doing less damage to the muscle when you're not pushing them very hard. So with the group that received placebo, those went down a little bit, as we'd expect. The group that got EDG 5506 had a significant decrease in both their CK as well as the fast troponin that shows that it is targeting the specific fibers that we're trying to target, and in fact, very close to normal levels. Earlier this week, I showed data at the New Directions meeting that if we have these same patients actually go on to take the drug in the real world, that we also see suppression of the CK and the fast troponin. So it's not only working in the setting of a phase 1 unit, but we're also seeing this protection in the real world when people are walking around and doing their normal activities. So we're very encouraged by that. So we are developing this, as I mentioned at the beginning, not only for Becker, but also for Duchenne, because and certainly we've heard this from yesterday at the Becker Forum from Dr. Apkin, that this is a spectrum of disease, from the complete lack of dystrophin to the partial lack of functional dystrophin in some patients with Becker. So we are looking at this over the disease spectrum, basically. And we know, and I would point out, of course, the posters are now down, but we had a couple posters looking at what Becker looks like in the Duchenne registry, who we worked with to understand how quickly people are going off their feet, and also other symptoms like muscle pain. So these are all things that we've learned about that we'll be measuring in the future clinical trials. So what our goal is to develop this drug as an oral drug to preserve, to improve function in Duchenne and Becker, and this is regardless of mutation. So potentially this could be used not only by itself, but in conjunction with other therapies. And it's very exciting to see a lot of different therapies that are being developed, but it's not clear that any one of them is going to push it all over the edge. And we're going to need more than one thing to be able to make the most difference in this disease. And basically, we're going at the mechanical stress-induced damage, which is really where this disease begins. So we have clinical trials both ongoing and in preparation. The ongoing open label study has enrolled all of the patients from the phase 1 and is keeping them on escalating doses of the drug for a 1-year study now. Getting a lot of information from that. There's also a natural history study that is being conducted by the GRASP academic network, and Edgewise is funding, and a natural history study of patients with Becker that are age 8 and above. And this will give us and the community important information about the disease and how it develops. We're doing that also in collaboration with Dr. Vandenborne and the Imaging DMD to understand more about the disease. We're beginning a study in adult and ambulatory patients that I'll tell you more about in the next slide that's planned to start in the coming weeks. And later in the year, we're planning an initial study in Duchenne to understand the safety and pharmacokinetics in boys. So the trial that we're planning in Becker, there we go, is going to be enrolling adolescents age 12 and up to an age of 50. Patients will need to have a confirmed mutation in the dystrophin gene, as well as a Becker phenotype, and that's defined as having been continuing to ambulate past 18. We are including ambulatory patients, not on corticosteroids. This will be a 12-month placebo-controlled study. We'll be looking at safety, biomarkers, as well as MRI fat fraction of the upper leg, since that, as you'll hear about, is an important way of assessing the disease in a very objective way. We'll also look at functional assessments, including North Star and the North Star assessment for limb-girdle muscular dystrophy, which is an expansion of the North Star for adults that has been used in limb girdles. So we'll be looking at that and other functional assessments. Lastly, I want to extend thanks. We really had a wonderful response from the community in enrolling these 2 studies, and we're very appreciative of all the sacrifices that participants and their families are making to be in these studies. So Thank you, and we'll have questions afterwards. Okay. This doesn't work. This doesn't work.
Our next speaker is going to be Dr. Paula Clemens from the University of Pittsburgh. He's going to tell us about a study with Vamorolone in Becker patients.
Hello, good afternoon. Can everyone hear me? Okay. And I really want to thank the Parent Project for inviting me to present. What I'm going to tell you about this afternoon is a study titled VBP15 BMD 001, and it's a phase 2 pilot study of Vamorolone versus placebo for the treatment of Becker Muscular Dystrophy. And good. So these are my disclosures, and below in the box is the funding for the project, which includes the NIH, the FDA, the Foundation to Eradicate Duchenne, ReveraGen, which is the sponsor that developed Vamorolone, and Santhera, the sponsor who is licensed Vamorolone. So as Joanne has introduced, Becker and Duchenne fall along a spectrum, and we most commonly see Duchenne muscular dystrophy when the mutation causes a loss of the normal reading frame so that you lose the entire right end of the protein, and we recognize that that results in a severe Duchenne phenotype. However, nature has shown us that it is possible to have an in-frame deletion in the dystrophin gene that leads to a truncated protein that's missing a segment in the middle, but retains its left and right end of the protein. And that can result in a milder phenotype that we call Becker, although Becker can cover the whole spectrum of severe to mild to even probably asymptomatic. So corticosteroids have been used extensively as a standard of care treatment in Duchenne muscular dystrophy, and multiple studies over now 15 or more years have shown that they can improve strength and mobility. They prolong the period of ambulation, and prolong arm function, prolong respiratory function, and have largely eliminated the need to have spinal surgeries because of their beneficial effects by preventing scoliosis. But steroids also come with lots of side effects that are listed there on the right, and we haven't typically treated Becker muscular dystrophy with steroids, although some Becker patients will be treated with steroids, but particularly in Becker where the lifespan is long, we know that there are really cumulative effects of steroids in adults, including actually very detrimental effects on muscle, ultimately causing muscle atrophy and certainly having very severe bone effects as well as metabolic effects. So we haven't typically used steroids in the treatment of Becker. And so one of the questions is, would it be possible to harness the benefits of steroids but uncouple them from the side effects so that there wouldn't be as many side effects? And that is what really led to the development of Vamorolone. So as you can see on the left, this is the chemical structure of Vamorolone, which shares a lot of features with corticosteroids, but it has some important differences that lead to pretty significantly different ways that it functions. And we call this a first-in-class dissociative steroid. It is not a corticosteroid. But like corticosteroids, such as prednisone or deflazacort, vamorolone is a very strong inhibitor of inflammation, and we think that's the main way that it functions in dystrophic muscle. It inhibits one of the main signaling pathways that lead to inflammation in muscle that go through a signaling molecule called NF-kappa B. So vamorolone retains that activity, but it has less of some of the other activities that we see in corticosteroids. So corticosteroids interact with glucocorticoid response elements, and that leads to a lot of the side effects that we see. So vamorolone has been shown in a lot of preclinical studies to have reduced activation of those glucocorticoid response elements. It also reverses the activity at the mineralocorticoid receptor and becomes an antagonist. And we know from other studies of other drugs, specifically eplerenone, that feature is beneficial to the cardiac disease in Becker muscular dystrophy. So there has now been a family of studies of vamorolone in Duchenne. And so there was first a phase 2A study in which there were 48 participants, age 4 to 7, with Duchenne muscular dystrophy. And it began with a dose finding to find the most effective dose of vamorolone, and then continued on into a 6 months treatment, 18 months, and 30 months. And shown there on the slide are the 4 publications that have come out of the results of those studies. And with those results and knowing the optimal dose of vamorolone, a phase 2B randomized controlled study was initiated again in Duchenne, age 4 to 7. There were arms for placebo and prednisone, as well as 2 doses of vamorolone. And all of these studies are now completed. And any participants who wish to, which actually was by far the majority, have continued on in expanded access protocols, continuing to receive vamorolone. So an overall summary of the studies of vamorolone in DMD has shown a functional improvement or stabilization for up to 30 months in the open label study, and a very large majority of the participants continued on with the study. And in fact, really the only participants that left the study were because they wanted to get onto corticosteroids to make themselves candidates for gene therapy studies. But overall, it was extremely well tolerated and safe. And there were statistically significant and clinically relevant functional improvements that showed similar improvements to prednisone and a similar separation from no treatment. But very encouragingly, vamorolone had fewer side effects, and even in just the 6-month study showed a preserved growth trajectory in treated boys. And those that started on prednisone for that first 6 months actually did have growth suppression, and when they switched to vamorolone for the second 6 months of the trial, it restored their growth trajectory, and they were growing normally. So I'm going to switch gears now and start talking about Becker. And there is a pair of PhD scientists at Children's National, Alison Fiorello and Christopher Heyer, and they have done really impactful studies, preclinical studies on Duchenne and Becker. And what I'm showing here is a Becker mouse that they created. So they essentially engineered an in-frame deletion in the mouse dystrophin gene. to create this mouse, and then they have really extensively characterized it. And so what you can see on the left-hand side of the slide is the Western blot that by this point in the conference I'm sure you're very familiar with of dystrophin, which is the sort of the upper bands here. And so here's a wild-type mouse with dystrophin, with its dystrophin bands. This is the Becker mouse, and you see that there is still dystrophin present, but it is reduced, and then the MDX, which has no dystrophin. And you can see this effect in two different skeletal muscles, the diaphragm and the triceps, as well as in cardiac muscle. And here you can see what you would see with in the Becker mouse on the left-hand panel here with just dystrophin, where there is many fibers that don't express dystrophin. Little blue dots are the nuclei, and the red is the dystrophin. But there is some dystrophin present, and we know that that's truncated dystrophin because of the genetic mutation in these mice. And when these mice were treated with prednisone, and even more so with Vamorolone, we see that there is a preservation of the dystrophin. So, and that's reflected in these histological sections where you see a much more healthy-looking muscle in the treated muscle. And then over here just shows the Western that shows that there's been preserved dystrophin. And that gets to a scientific concept that we're familiar with, which is that there are many factors in muscle that, that will degrade dystrophin. And one of the main mechanisms that does that is the so-called microRNAs. And these are inflammatory molecules that bind to the truncated dystrophin that's produced in Becker muscle and degrades it degrades the actual protein. So that we believe that by inhibiting some of these inflammatory pathways in muscle, we are actually preserving the dystrophin that is expressed in Becker patients. So getting to the BMD study with Vamorolone, I'm showing here some of the key inclusion factors. So this study has eligibility to males with confirmed Becker diagnosis by genetics and clinical presentation. The study is going to be exclusively in adults for this first pilot study, and we're targeting patients who are affected but still ambulatory. The typical exclusions are just sort of chronic illnesses or severe heart disease that could make it unsafe for a participant to participate in the study, as well as, of course, being on steroids or those agents that trigger the mineralocorticoid receptor, since these are factors that we're studying in the study with Vamorolone. So in this study, Vamorolone will be given as a 500 milligram daily dose. In the children, it was a milligram per kilogram dose, but typical for adult studies, this is a fixed dose, and it's designed based on the studies in Duchenne boys to be an effective dose. It will be versus placebo, and it's a 24-week treatment trial. So of course, as any early phase study, a major objective is to determine that it's safe and tolerable, but we have every reason to think that it will be well tolerated and safe, as it was in healthy adult males in an original phase 1 study as well as in patients with Duchenne. We'll be looking at the pharmacokinetics, which is just how the drug is excreted, absorbed through the GI tract into the blood and then excreted. And we're also looking at a pharmacodynamic approach of looking at serum factors that can reflect some of the safety concerns that one might have, as well as some efficacy markers. Although the clinical efficacy is an exploratory objective, again, because this is an early phase trial, it is a very important objective and will be studied throughout. And of course, patient-reported outcomes. Now, there will be an optional muscle biopsy, and these will be done as needle biopsies before and after the treatment period. And here we want to specifically look at dystrophin induction in muscle, and the laboratory who studied the Becker mouse, Allison and Chris, will be studying the dystrophin as well as the microRNAs. And this is really important because of our, from the preclinical studies, thinking that this will preserve Becker dystrophin. We will have an opportunity to test that. So the study design is shown here. There will be a screening period where eligibility is determined. And then, whoops, there we go. These are demonstrating the muscle biopsies, both before and after the treatment period. The treatment period will be for 24 weeks. And the randomization, which will be blinded, will be 2-to-1. So 67% will be on Vamorolone, 33% on placebo. And at the end of the 24 weeks, there will be an opportunity to enter into an expanded access program if participants wish to continue receiving Vamorolone, or they can enter into a tapering period if they wish to come off of it. So I will bring my presentation to a close. This shows many of the foundations and granting agencies that supported the early development of Vamorolone, and including the parent project of muscular dystrophy. And this was critical so that this could be developed in kind of a venture philanthropy model. Really, it was all of these contributions that made this a reality at the early phases of this. And as I mentioned at the start, the industry sponsors are ReveraGen and Synthera. This is also a Synergy study. Synergy is a cooperative international neuromuscular research group that has been, since the year 2000, conducting some multi-center academic network that has been conducting trials in muscular dystrophy. The two enrolling study sites for this pilot study will be the University of Pittsburgh and the University of Padova in Padova, Italy. And I think that is, and one very important factor of this study is that we absolutely need to recruit participants who are outside of Pittsburgh. And so what I've shown here is I have outstanding study team with Gabriella Nizawa and Kathy Van Every. If you are interested in participating and you have our contact information there, if there's anyone that you can share it with, I would be more than happy. We have a sort of physician flyer that I would be happy to send to you or to give to your physicians or to send to them directly. So please don't hesitate to contact me, and we will be really happy to hear from you. All right, thank you.
Thank you, Dr. Clemens. Our final speaker for this session is Dr. Krista Vandenborne from the University of Florida. Is the PI on the imaging DMD study and is able to share about some of their work with Becker.
Good. Good afternoon, everybody. It's a real pleasure to be here back at the PPMD conference in person. It just feels really good to see so many familiar faces. I would like to thank the PPMD organizing committee for organizing this special session on Becker's muscular dystrophy. And I'm looking forward to share with you some of our more recent work, which focuses on the development of imaging biomarkers for Becker muscular dystrophy with the goal of accelerating therapeutic development. So, as many of you know, the process to move a drug from testing it in cells or animal models through these different phases of clinical trials is often a very long process. And there's a number of reasons for that. But one reason that we have really focused in on is the need for let's see. Should I be using this one? The need for sensitive outcome measures that can really quickly detect whether a drug is working or not, and that can do that in an unbiased, objective manner. So our team has really focused on the development of magnetic resonance imaging outcome measures in the context of muscular dystrophies. We've been very fortunate to be funded by the National Institute of Health to really develop biomarkers, and we are completing a large multi-center natural history study in Duchenne muscular dystrophy. Over the last 15 years, we've been able to collect a lot of longitudinal data in boys with Duchenne, thanks to the help of many of the families that have traveled to one of our sites. And so we're now really in a position to leverage that knowledge and the information that we've gained and the data to help with MR biomarker development for Becker muscular dystrophy. So the goal of our study now is to characterize the natural history of the disease progression. You already heard earlier from Dr. Donovan the importance of that. We really need to know what the natural history is in order to interpret the effect of drugs. So we're going to do that in a number of skeletal muscles. And we're going to develop sensitive MR measurements that are clinically meaningful and that can be used in the context of clinical trials to either allow us to do trials more quickly or in a smaller number of patients. And that's really important if we want to move forward. For the purpose of my talk today, I split it into two parts. One, I'm going to share with you some of the key lessons that we've learned from doing MRI in Duchenne boys. And then secondly, I'm going to share with you some of the initial results in Becker's. So first thing that we learned is that it is very easy to detect muscle pathology in muscular dystrophies using MRI. So here you have two images. So on the left side, you have an image taken of the upper leg of an unaffected subject. And on the right side, you have the same type of image taken in a child with Duchenne that's 12 years old. So typically, if you look at the muscle here, these are the quadriceps muscles, the signal is quite homogeneous. And that's simply because the muscle fibers are nicely organized and you have this kind of smooth gray signal intensity. On the other hand, if you look at the image in the child with Duchenne, you can see that there's a lot of structure in the muscles. You can see all of this bright signal, which is really caused by the replacement of muscle by fat. So we've put a lot of effort into quantifying this fat infiltration. And so here on the next slide, you'll see how we do that. So to quantify the fat infiltration, we measure something that we define as muscle fat fraction, which is essentially the fat over water plus fat. And you can do that using MRI by taking two sets of images. So here you have a water image, here you have a fat image. And if you combine these images, you have now a quantitative fat fraction map. And each of the pixels in this image is now a true measurement of fat fraction. So based on that, now you can go back and you can measure the mean fat in a muscle or in muscle groups, a number of combinations you could potentially consider. So this just shows you how important fat infiltration is when we're looking at disease progression. So these are images that are taken in a young boy with Duchenne muscular dystrophy, starting at the age of 8 years old. These are now images taken of the lower leg. So just to orient you, these are the calf muscles here with the soleus, which is a muscle that we have measured very carefully. In this type of image, fat or lipid is shown as green, and water is really mainly muscle. So if you look at when the boys are young, so below the age of 10, you can see that the muscles are pretty well preserved, but you could start seeing some fat infiltration here in one of the calf muscles. Once the boys are older than 10, you can clearly see the wasting of the muscle and the progressive replacement of muscle by fat. So fat infiltration or muscle fat fraction really tracks disease progression. And that's very important. We're going to look at drug interventions because we can measure whether that fat infiltration happens in the same time course or not. So now we have quantified muscle fat fraction in a large cohort. This study was completed here in a total of over 100 boys with Duchenne at different ages. So here we now have fat fraction measured in one of the quadriceps muscle. This is the vastus lateralis. And we've displayed this for different age groups. In yellow, you have the control healthy subjects. And you can see that fat fraction is really low. It's around 5% or less in healthy muscles. However, if you look at the boys with Duchenne, at baseline, you have the light blue here. You can see, of course, the increase. And then you can see in dark blue, the same boys that were measured 12 months later. And you can easily detect the changes over the course of 12 months. And again, most of the drug trials are done in 12 months or 18 months period. So this time frame is really important. On the right side now, we have the same kind of data, but this shows you individual data. So again, fat fraction as a function of age in the young boys with Duchenne. Each data point or each circle here is a data point. And so what you can see is that you have a slow progression in the beginning, then a rapid phase, and then again, kind of leveling off as the muscle is replaced by fat. You can also appreciate that there is a lot of variability between the boys. Some of the boys, this happens much faster than other boys. We've also seen that this trajectory varies across different muscles. Now, in boys with Duchenne, this is in a pretty typical order. We kind of know what that order is. We don't know that yet for Becker's. So here what you could see is that in the upper leg muscles, such as the biceps in green, and here the vastus, which is one of the quadriceps, this happens really fast. In other muscles, such as the forearm muscles, this happens much slower. We've also demonstrated that muscle fat fraction is responsive to corticosteroids. And this is really important, right? We need to ultimately be able to detect the effect of a drug. And we've heard earlier that in boys with Duchenne, corticosteroids have been shown to delay loss of ambulation by about 2 years. So as a proof of principle, we've looked at the effect of corticosteroids on muscle fat fraction. We've looked at that both short-term and long-term. So here you can see the 1-year change in muscle fat fraction for the two muscles, the soleus and the vastus lateralis, for the boys that remained corticosteroid naive, and the boys that went on corticosteroids. And you can clearly detect the effect of the corticosteroids. You can also appreciate the difference in this disease trajectory between the boys that are corticosteroid naive, so in red, you can see a much faster trajectory versus the boys that were treated with corticosteroids. Again, just to demonstrate that these measures are responsive to the effects of treatments. Now, from the FDA's perspective, it is not just important that the measure is sensitive. The measure has to be clinically meaningful. So what does that mean? So what we need to demonstrate is that these MR measurements both correlate with functional endpoints and that they predict changes in function. And so in order to do that, at each of the time points that we had the boys visiting us at our imaging sites, we also asked them to perform a number of tests, of functional tests. And so we asked them to do the 10-meter walk-run, they climbed stairs, get up from supine, and performed the 6-minute walk test. And so what we found is that muscle fat fraction, as shown here, significantly correlates with performance on each of those functional tests. We also found that MR muscle fat fraction predicts both changes as well as loss of function 12 and 24 months later. So here what you have is the proportion of boys that could still complete either getting up from supine, climbing stair in red, or ambulation as a function of the fat fraction. So for certainly over the last decade, a lot of data have been collected in boys with Duchenne that have really shown the value of these MR measurements. They're also now being utilized in a number of clinical trials, and we've listed some of the clinical trials that are using MR measures. And so we really feel like we're in a good position to move this technology to help patients with Becker's muscular dystrophy. As you heard earlier today from both presentations, there is now a number of drugs that are in the pipeline for Becker's muscular dystrophy. And so we really want to make sure that we understand the natural history and that we have good tools available to help these studies. So at this point, we are expanding to collect data in men with Becker's muscular dystrophy. This study is being done at 2 different sites. The University of Florida, we're located about 2 hours north of Orlando, and then our colleagues at the West Coast, the study will be done at Oregon Health Science University. So as you heard earlier, it's really a spectrum. Duchenne and Becker sit on a spectrum. But in general, we know that Becker's muscular dystrophy progresses much slower, which means that the measurements that we use have to be even more sensitive than what we need in Duchenne. The population is smaller, which means recruitment is more difficult. Again, really putting more of a burden on making sure that we really have good tools in place. And as you'll see, there is even more variability in the disease trajectory. In conjunction with the MR measurements, we're also going to be implementing functional endpoints to help define the best potential functional endpoints. And this is being done in collaboration with GRASP as well as with Edgewise. So at this point, we have collected initial data on 28 men with Becker's muscular dystrophy. This shows you here the overall demographics, so average age of 39 with a wide range between 18 and 62. 5 of the 28 men that were enrolled were non-ambulatory, and the average performance on the 6-minute walk test for those men that were ambulatory was 370. This shows you our inclusion criteria here as well as our exclusion criteria. So just a high-level overview of the functional status of the 28 men that we studied. On the left side here, we displayed the scores on the upper extremity Brook, which essentially has 6 different grades. We found that the majority of men had a score or a grade of 1, which is highly functional, so that meant they could still abduct their arms in a full circle until they touch above the head. For those of you that are interested in the PUL score, which is another measure of upper extremity function, the average PUL score is listed here as a 37. When we looked at the lower extremity muscles, you could see we had a much wider range, and the majority of men were able to walk, but they needed aid of a railing to climb the stairs. 16 of the men could not rise from the floor, just to give you a perspective of the population that we've studied so far. The score on the NSAD score is listed at the bottom here with a wide range of values. So next I'm going to show you some of the images acquired in the different muscles. So these are images acquired in the upper leg muscles of 6 ambulatory men that were enrolled in our study. All of the images that I'm going to be showing are going to be organized in the same way so that you can easily see from one muscle group to another muscle group. So here on these images, on the top left, we have a 21-year-old individual that really had minimal symptoms, mainly muscle pain and muscle cramps, but you can see a very high score on the NSAD and Brook. And what you can see here, these are images now again of the upper leg with here the quadriceps and the hamstrings, very little fat infiltration. On the other hand, when you look at some of the other men, you can see that there is significant fat infiltration in the upper leg muscles. Some variability, but overall we found that in the upper leg muscles of these men, there was a lot of fat infiltration. If we look now at the lower legs, so remember these are the same exact men I'm going to show you. So these are now the lower leg muscles. So overall you can see more preservation of the lower leg muscles. Couple of points that I want to make is that there is significant heterogeneity. In general, for most of the men, you can see that the gastrocs, so the calf muscles here, that there is a lot of fat infiltration. But the other muscles are more variable. For instance, you could see in the soleus, it goes across the different ranges. Here you have a gentleman where the tibialis anterior is very infiltrated, but the soleus is not. And here, this gentleman is almost the complete opposite. Just to point, there's a lot of variability, so across the different men that we studied. Next, I'm going to show you images of the arms now. So these are, again, same exact men that I showed you earlier. So here you could see, again, the gentleman had minimal symptoms. Here's a 28-year-old with very high upper extremity function, and you can also see minimal fat infiltration. We had a number of subjects that showed nicely preserved triceps, but a lot more infiltration in the biceps. This shows you now the group results for all of these men. So overall, we saw that the upper leg muscles, you can have the hamstrings and the quads here in dark blue, had the highest levels of fat infiltration, as you saw on the images. We saw that there was significant fat infiltration, but around about 30% on average in the arm muscles, and here are the values for the lower leg muscles. Now remember, even though I give you those average values, there was a lot of variability. So one gentleman may show high fat infiltration in one muscle, but not the other muscle, and the other way around. So that's always a challenge when you're developing measurements when there's that much variability. So what can we do with that? One approach is that we come up with what we call a composite measurement. That's a measurement that takes into account all of the muscles. And so that's what we're pursuing right now. So on the next slide, I'm going to show you a composite fat fraction across all of these muscles. And this is here a figure showing the relationship between the performance on the NSAD score, so one of the functional measurements, and the composite fat fraction. And you can see that there is a very strong association. So the men that had low fat fraction values scored very high on the NSAD. And then inversely, the men that had high fat fraction values scored very low. So that's one approach that we think is going to be really valuable in the future, and that we will need to look at a combination of muscles and not a single muscle like we've done maybe in Duchenne. We've also looked at the responsiveness to treatment, and this is a study, this was a phase 2 clinical trial that was completed by Ital Pharmaco, looking at the effect of Givinostat that was completed last year. And they looked at fat fraction, in this case, in the whole thigh, as well as fat fraction in the quadriceps. They found that even though there was no change in fibrosis on muscle biopsies over 12 months and no change on the performance on these timed tests, They found that muscle fat fraction in both the thigh and the quadriceps, one, changed significantly over 12 months, but more importantly, they found a difference between the placebo group and the treatment group using these MR measure, just providing some initial evidence that this may be indeed a sensitive measure that could be considered in the context of clinical trials. We've also started looking at respiratory muscles. We know from the literature that there is potential for BMD patients to be at risk of developing respiratory dysfunction. And so at this point, we are looking also at respiratory muscles. Ideally, we want to measure the diaphragm, but it's a very thin muscle, and it moves while you breathe, so it makes it really difficult. So we're taking a very different approach, and instead we're looking at accessory respiratory muscles. So here are, these are now images taken through your thorax, kind of at the level of your belly button here. So here's your belly button, these images, here's the vertebrae. And so we are looking at these muscles here. This is the external oblique and the internal oblique. So these are muscles that help during breathing, and particularly during expiration. And so here on the top, we have again images taken of an unaffected control. So you typically should see 3 muscles here. So this is the transverse abdominis, the internal and external oblique. If we look at the individual here at the bottom with Becker's muscular dystrophy, you can see fat infiltration, in particular in the internal oblique muscle. When we looked at all of the subjects, we really saw a lot of heterogeneity again, with some of the muscles preserved. So here, for instance, in this gentleman, you can see that these muscles show very little fat infiltration. We had about 13%. Other individuals had higher values, and we had one individual whereby you saw very significant fat infiltration. So quite variable, but a little bit higher than what we had expected. So here are the summary results for all of the subjects that we have studied so far, showing you again the range of muscle fat fraction in these 2 muscles, really showing a very wide range. If we compare that to the leg muscles here, so we added these in, you can see that the respiratory muscles had significant fat infiltration, and we were a little bit surprised to find that. So we have started to look at the value of these measurements. Unfortunately, due to COVID, we have not been able to do a lot of pulmonary function testing, but we do know from the work that we've done in Duchenne that these measures are valuable and that they progressively increase, similar to what we've seen in the other muscles. I'm just going to finish out here with this slide we heard earlier today from both Dr. Donovan and from Dr. Clemens, of the importance of inflammation and damage, particularly earlier in disease. So at this point, we are developing methodologies to look at inflammation and damage in young BMD patients. So these are images here taken in a 12-year-old boy with Becker's. This is a water map, shows very little. Here you can see a T2 weighted water map, and essentially what you see is these lesions here show evidence of muscle damage and inflammation. And we think that this approach may be particularly helpful with some of the anti-inflammatory treatment strategies or the treatment strategies that are proposed to reduce stress on the muscles. So I'm going to finish here by thanking everybody. I want to first of all thank all of the patients and all of the families that have traveled to one of our sites to collect all of these data over so many years. It's really thanks to all of the patients that we have gained so much knowledge and learned so much about the disease. And I also want to thank our funding agencies and Parent Project Muscular Dystrophy. Thank you.
So at this time, we'd be happy to take any questions from the audience. If you want to raise your hand, I think Rachel's running around with the mic right now.
Just a quick one. Do any of you know if there's any plans to do studies in non-ambulatory Becker patients? Like treatment studies?
So I may take that first. I would say that we are looking at a wide range of ambulatory patients, and that ultimately we want to look at the broader range of patients, but we don't have that in the immediate future in the next couple of years, I will say.
Yeah, I think that's probably pretty typical, and in that usually the initial studies are going to be in patients who are ambulatory, but then because that is really the group that there would be the most opportunity to show a benefit, if there is one. But then that would then, if that is seen, then that would open the possibility to expand it to a wider range.
This is a somewhat prosaic question about Vamorolone. Many of us old-timers recall that we used to buy deflazacort generically from Europe, and it cost me about $1,300 a year in the case of our child. Mm-hmm. And then a US company got it licensed by the FDA, and the price went to $89,000 a year. And even though it was subsequently sold off to some other company and the price has been reduced, my child still reports annual rows with Blue Cross Blue Shield over the pricing of it. So is there any theory about the pricing of this, or is this going to be something else that's constantly a problem?
Well, I would agree that it's a problem. Of course, I'm an academic and a physician, so I spend a lot of time. I greatly preferred when families got deflazacort from other places, because now I spend a lot of time, arguing for why it's important and with insurance companies. But yeah, it's a very unfortunate part of our, and I think that that was a big part of why Vamorolone was developed through venture philanthropy, so that ideally it wouldn't have to go to a very high cost. I think ultimately at the end of the day, to market a drug, you have to have a pharmaceutical company, and so it probably won't be kept, it certainly won't be as low as prednisone. At least the academics that are involved would really like to see the costs not skyrocket. But I definitely, I totally understand your pain on this. I think it's really unfortunate.
I have a question for Dr. Donovan. The drug that you described that you guys are producing at Edgewise, it has a really interesting and kind of unique mechanism of action in that it's very selective for fast myosin and inhibiting that. It seems like it would have potentially broader applications beyond just Duchenne and Becker to other muscular dystrophies too. But the thing that makes me wonder, because it's so selective, it wouldn't have an impact on slow myosin or on cardiac myocytes. And with some of the thinking about workload on cardiac muscle, and by preserving strength and motor functioning, one might actually negatively impact cardiac function. It makes me wonder what you guys are looking at in these planned trials for your compound, and how that would be addressed as far as the possible impact that could be negative on the heart with this drug.
Yeah, that's an important question. I think that first, to the slow muscle fibers, there's important crosstalk. So when you have inflammation in any fibers, you've created a negative milieu that inhibits replication of stem cells and restoration of myocytes. So in the animal models, because all of the muscles basically have a mix of fast and slow fibers. So in the animal models, we do see an overall benefit. It's not that only the fast fibers benefit. And what we see is a decrease in fibrosis over the long term in the more severe MDX, the DBA mouse. And interestingly, in the heart, we also see a decrease in fibrosis. This is despite that the drug doesn't specifically impact the cardiac fibers. But as you know, the interplay between respiratory and cardiac, we know they have an effect on each other in the clinic. There may be some crosstalk that is positively impacting the heart as well. So we certainly don't see any evidence of a negative effect, and we do see some evidence of a positive effect. Now, that being said, in the clinic, we are following both pulmonary function and we are following cardiac echo as well in these patients to look at function.
We did have a question that came in online for Joanne. What is the age range for the study, and do the boys have to be on, or sorry, are they allowed to be on steroids?
So we are not including individuals that are on steroids at that time. I think that, as was alluded to before, it's to make the trial more homogeneous in terms of the population to make it more sensitive. So they won't be able to be on steroids. The trial will include age 12 and up. And what I didn't mention is that we are looking to have a range of North Star values at baseline from 10 to 32. And the reason for that is that patients who are at the highest level of North Star then tend to remain there. And there's a lot of good data from Padua, from Professor Luca Bella, that the sensitivity of North Star as a measure in Becker is improved if you look at this kind of a range of patients between 10 and 32. So they have indeed started to decline. So the lower end of the age spectrum is 12. The upper end of the age spectrum is 50. And that's because when you look at that kind of a natural history curve, the patients above age 50 tend to be those that are ambulatory, tend to be ones that are continuing to stay at a high level of North Star. And so to make the trial more sensitive, that's why we have designed it that way. I mean, ultimately, the intent is to show benefit and to have the drug be able to be used in a broad range of patients. But in order to make the trial as likely to succeed as possible, that's why we narrowed the range to improve how much signal we can see, how much change, versus how much noise that obscures it.
I don't see any other hands up in the audience, so I think with that, I'm going to thank all three of our speakers for sharing updates on these trials with us today. And I've also been told that as we conclude this, if you can all head out towards the glass doors at the end of the hallway, I think we are gathering for a group picture. So with that, I'd like to thank our three speakers. So.